Not just sure, sure. It wasn't very clear. We don't have much of that, I'm sure. But like, it's really happy with you.
Thank you for giving all your insurance.
No, same, yeah.
If I were to go to any kind of accident, yeah, for it, yeah.
All right. Got lab today. So we got a couple presentations. I got yours. So I don't know if we might only have one presentation today, but we'll get them up. Did you email me? Yeah, you did. The audio. We'll get that up. The exam, I've gone through, checked some answers, checked all of the answers that had asked for a genus, identification of the genus. So, yeah, if you gave me genus and species, if you gave me a SPA at the end, those were all marked wrong, so those got all corrected. I then also ran some item analysis to look for questions that were like all marked wrong. And there were a couple across the two lecture exams where the wrong answer was selected as being correct. So I updated it, ended up regrading. It should be accessible, so you should be able to check like the key, check your answers. I looked through probably 10 to 15 different questions between the life cycle part and the regular general question part. And there were, I think, 3 or 4 where the key was wrong. So those were fixed. If you think you found another one, please email me. All right. Don't tell me number, I think number 46 is wrong because I don't know what number it is. You're going to get a different order if I cleared it and you could get a different question. So just tell me, what the question was asking, what you answered, and then I should be able to find it and see what the key looks like. In some cases, it might be something where I would tell you, check, look at your notes, look at this presentation, think you'll find the answer. Other times it might be, yep, sure enough, I had the wrong answer selected, so we'll get that fixed. So go ahead and check it. I kind of messed around with the grade, the total points on that life cycle. So I guess I can set the total points down to like 20 points instead of being at a total of 30. And it changes like the percentage that it shows. I don't know how it would change the overall grade calculations. So I've just left it at 30, but know that whatever you have, that's out of 20, but we're not doing any of that extra credit. So if you had more than 20 points on that life cycle, you got a 20 out of 20 on those. All right. So that kind of keeps it in line with previous years where you had a choice of which life cycles to diagram. All right. All right. And I was working on the quiz. I got sidetracked. We'll get those up. We'll get those up. We left off with Trichinosis, whipworm. All right, we did our life cycle. So life cycle, it's in the large intestine, right? Our worms, J3s, come out. They have to penetrate in the crypts of Livercune. They penetrate actually at the base of those cells, or at the base of those crypts, and then they make their way back to the luminal surface, where along the way they're molting, they are growing, and ultimately forming this epithelial syncytium that helps to lock the worm in place. And we said that this whipworm, it's associated with humans. That's who it infects. Now, there's other whipworm species out there that goes into other organisms, but Trichinosis is humans. We find highest prevalence in those areas that are warm and moist because those eggs are susceptible to desiccation. They need to be kept moist. And the warm environments just speed up development of that unembryonated egg. All right. Also, places where geophagy is common exhibits higher prevalences, and those places that utilize human waste also tends to have higher prevalence. Now, do we use human waste here?
No,
What's that?
I don't think so.
You don't think so?
You have to use it?
Yeah.
I mean,
Humans, do we use human waste as fertilizer?
I thought we used it more cow.
I've been calling in
probably would.
like actual business and like the big farms and everything, they probably can't, but it would start with people still do.
So there was a story out in California several years back. Say a decade back. I believe I was here. It came like that first year I was here. People were complaining that they had tomato plants growing in their yard in the grass. People were like, "What the heck's going on here?"
Were people eating tomatoes and then using their waste in the garden and then just
So-
tomatoes were growing from that?
Those people weren't. So what do you know about sewage treatment plants? Sewage treatment cleans water, right? Right? What happens to all the solid wastes? They do break down, but they don't disappear, right? So at sewage treatment plants, they have this black solids that they would then kind of pour out and utilize as fertilizer. You can, for the most part, it's safe because it's gone through, the entire process. It's almost like no different than going out into a wetland and taking out, the silt that is right down at the bottom. In a lot of ways, that's what it is. But yeah, the tomato seeds were surviving. Some other vegetable seeds were surviving the treatment, and they're in this black thing. I bet if we went here, we would see it. I had a aquatic ecology class in grad school, and one of our field trips was to go to the sewage treatment facility out there, and they do. They've got an area where they kind of dump it out and it dries, and then they can break it up and they'll sell that as fertilizer, kind of like horse manure. But would whipworms survive that? No, not at all. With this night soil, we're talking about basically fresh soil, fresh waste. So bedpans, right? Bedpans. All right, so what about disease and pathology? So if you get it, if you exhibit, you have trichinosis or whipworm disease. So I put up both to cover our bases. Now, most of the time, this is asymptomatic. You'll have it and not really know you have it. This is especially true if you have a light infection, so less than 100 worms. As you have more than 100 worms, as you start getting heavier infections, then you can start showing some cases significant pathology. All right, what sorts of things are you going to see? Well, dysentery is one. So dysentery, is basically explosive diarrhea. That's your colon. What's the function of the colon, the large intestine?
To get the last bit of nutrients possibly can.
Water too, right? Yeah, water reabsorption. So you've irritated the intestinal lining, I should say. The worm has irritated the intestinal lining, inhibits that, so you don't get that reabsorption. In some cases, it actually causes water to move out of the cells into the colon. So dysentery, you can get anemia. This would be iron anemia because the worm, as it's burst through, causes slight hemorrhages, small hemorrhage. And in some cases, it's just constantly hemorrhaging. Not a whole lot. I mean, it's not like it's dripping blood, but it's a very small amount of blood. And if you have, let's say, 500 worms, well, that's 500 tiny drops, which can now be, and that's per day, that could become significant loss. Growth retardation, inhibition of cognitive function. Those are both tied to, loss of blood plasma, loss of iron, toe and finger clubbing. If you've seen this is kind of roundness of the toes and roundness of the fingers. And you can think about or ask the question, well, hold on, why is this happening? Well, this is more of a systemic nature of the disease, of the infection. Yeah, the infection's is in the large intestine, but it's having such an effect on the body that you can start developing this toe and finger clubbing. Now, if you get rid of the worm, this goes away. This is a temporary case. Same thing with the anemia and the dysentery. Prolapse rectum, that's awful. That's awful, but again, it's severe inflammation, severe irritation in the rectum, in the large intestine, will actually cause it to prolapse out of the individual. And in some cases, death is possible. And that's just kind of like your body's been fighting it. It can't take anymore, so it starts to shut down. Fortunately, most of the cases, though, are asymptomatic. All right. Questions? All right. The next worm. So again, these are the stichosome possessors. The next worm that we talk about is Trichinella spiralis. Who's heard of this worm? I think most, if not all of you have. All right, this is the smallest nematode parasite of humans. So only 1 to 3 millimeters in length, and that is the adult worm. So it's the smallest nematode parasite of humans, but it's also the largest intracellular parasite of humans. Now, again, think about that. You can say that whipworm is intracellular, it's burrowing through, but it's burrowing through multiple cells and forms a syncytium. This worm actually infects a single cell and remains inside that single cell. So it, at least during part of its life cycle, it is an intracellular parasite. Now, this parasite has a rather unusual life cycle. And the reason it's unusual is that the definitive host also serves as the intermediate host. You typically don't see that. So, what's the life cycle? This. Apologize to anyone who is watching. So we move the camera. All right. So we're going to start inside of our definitive host. This is Trichinella spiralis. Vertebrate, it goes into numerous vertebrates. All right, so humans go into humans, pigs, small mammals. There's a lot of things serve as a host for them. All right, so our host will consume a nurse cell, something that's called a nurse cell, which has our J1 inside. All right, so we'll come back to what a nurse cell is in a bit. J1 gets released and is now in our intestine. Small intestine normally is where it gets released. All right. And then our J1 is going to penetrate out of the gut. Okay. And it goes into the gut mucosa. So I'm going to add some other stuff to kind of get to our locations. So it penetrates into the cells where we are now inside the gut mucosa, and it develops into a J2, J3, J4, and ultimately the adult. Right? Ultimately the adult. This is all in our gut mucosa. Right, so it's in the lining of the gut, in the cells. And this is burrowing through the cells, kind of like Trichinosis. Right, so it doesn't go into one cell and stay. It's going to just burrow through, consume cytoplasm, host cytoplasm. All right, utilize that for food source, grows, develops, gets to the adult, where the adults find each other. They're going to mate. And then these adults, or the females, are going to release our J1s. So, it's birth and escape. So this is a parasite that is ovoviviparous. Alright, so our J1s are juveniles, I should say, are embryos, ultimately J1s. They're developing inside of an egg membrane in the uterus. Alright, but they're not born in an egg. Right? It's not a shell. It's just the membrane. So the J1s are going to be born, right, out of the uterus, and then they're going to escape out of the gut mucosa. Once they escape, they're going to enter the circulatory system. They're going to get into the circulatory system, where they're going to travel through various parts of the body. And they're going to escape that circulatory system and go into various organs, various cells and tissues. Some of those, it's just the worm's lost, can't find its way. But other times, it actually finds where it needs to go and penetrates into that cell. And that cell we are talking about is skeletal muscle. Skeletal muscle cell. It goes into, smooth muscle, nothing. It goes into cardiac muscle, nothing. It's just burrowing through, right? But if it gets into a skeletal muscle cell, it's striated. Now, some interesting things happen. So we penetrated, we have this J1 now inside of a single cell, where it then induces developmental changes. It's developmental changes of the cell itself, eventually becoming a nurse cell, which has our J1 inside of it. Now, this change that happens, so you can think of it as this parasite gets inside the cell and then commandeers it, takes it over. All right? It changes the function. It's no longer going to be functioning as a skeletal muscle cell. It's going to be functioning as a nurse cell. What do you think the nurse cell is doing? Yeah, so just based on the name, it's going to basically nurture this J1 until it can get into our next host. So it stays here. It stays right there in the host. There's no connection because this host consumed a nurse cell. The parasite got out, developed to the adult, mated, then they release the J1s. Those J1s get into the muscle cells of that host, and now that host is serving as our intermediate host. So it served as our definitive host during this part of our life cycle, and now it's serving as the intermediate host. Right? Eventually, these adults will die. They don't live, for years. Those adults are going to mate, say weeks to months, and then those adults die. But you still had all these J1s that went and formed these nurse cells, and now that host is going to be the intermediate host. If the host doesn't get consumed, dead end for the worm. Right? The host gets consumed, and that's great. Right? The worm continues its life cycle. Now, there are some things that are notable about this life cycle, especially with the worm. Our worm, once it escapes from that nurse cell, switches to aerobic respiration. What is aerobic respiration? It's going to use oxygen for ATP production. Right? So aerobic respiration is all about creating energy, ATP. Aerobic respiration utilizes oxygen, which means if it's switching to aerobic respiration, at some point in its life cycle, it had to transition to anaerobic respiration. our worm is still perspiring, still surviving, still producing energy to keep it to remain alive. But when it's inside that nurse cell, it switches to anaerobic respiration. And I put it up here because we're going to talk about this nurse cell and talk about why it switches to this. Kind of an important feature of our nurse cell. Questions?
So this might be a stupid question, but does this mean that the J1 is the infective stage?
Yeah, the nurse cell inside the J1.
Okay.
Or nurse cell with the J1 is our infective stage. So if I ask what's the infective stage? Nurse cell. What's inside the nurse cell? J1. What's your headline? Diagram. Just cytoplasm on there for the gut mucosa. It's in the cytoplasm of the cells. It's burrowing through the cells, and I believe I have a diagram of it, or a conceptualized version of what the worms are doing. It's feeding on cytoplasm. So I did put some images here. Do have some images up here. We have, I think, female worms is all of the worms that we have in the lab. And you can see it. And you can definitely make out different structures of that female worm. All right. Males have this. It's a bursa, but the bursa looks like it's like a it's a V-shaped wing. You can say. But note for our female worm, we actually have our sticosome here, sticocytes. And it's those, we said the sticosome or the sticocytes have a different function based on what worm we're talking about. So Trichuris, those cells are used for the formation of that epithelial syncytium. For these cells, and I've got it on another side, they're going to be used during the formation of this nurse cell. So in our nurse cell, we have our worm penetrating into the skeletal muscle, and it basically reshapes the inside of it, and in some cases the outside of it, to form this cell, this nurse cell, where the worm stays coiled up and then just hangs out until our host gets consumed. All right, so the host depends on the type of life cycle. Yeah, kind of type of life cycle. I guess I probably should have said host depends on like species of Trichinella. Right? But they have kind of broken down life cycle types. All right, and that's kind of why we have it here. So the domestic life cycle, that's going to use it. So our humans can come in for all of these. We can be infected. But for the domestic cycle, it's basically humans, pigs, and rats. That's who's serving as our hosts. So if we live around rats, if we live around pigs, there's a chance we can pick up Trichinella. Especially if you undercook your pork, right? If you don't cook the pork sausage appropriately, you can get it. If you decide just to smoke, do like a low smoke, a lot of times that low temperature smoke isn't going to kill it. Right? But and in terms of us, because again, the cycle's probably pigs and rats. So in a farm type setting, you're going to have rats out there foraging. And occasionally a rat's going to die and then get consumed by a pig. Or occasionally some of the pigs will die and the pigs will actually consume them. Pigs are omnivores. Right? The sylvatic temperate, the sylvatic torrid, and the sylvatic frigid is all referring to the life cycles that involve wild animals. Right, so the domestic is all, say, farm raised. Sylvatic is our wildlife. So Trichinella spiralis can exist in the form of a domestic life cycle and sylvatic temperate life cycle. So our temperate life cycles more of moderate warmth areas, or it's our temperate regions. All right, but it's more than just Trichinella spiralis. So we have these other Trichinellas bertovi and Trichinellas morelli that we could become infected with. All right, how how do we get infected with them? Well, we're going to have to consume some wild animals. So typical hosts for that type of life cycle pattern is carnivores and scavengers, because the carnivores are going to feed on other individuals that are serving as our intermediate host, and then they themselves become intermediate hosts. And when they die, then a scavenger can come around, pick up the nurse cell, and keep our life cycle going. Trichinella bertovi is basically Europe and Asia. Trichinella morelli is here in America. So that's probably going to be the species. If you find them, you're going to probably find morelli. All right. Sylvatic torrid is super dry conditions, almost like your desert-like conditions. This is mostly in Africa. All right. This species is Trichinella nelsoni. You don't find spiralis out there. And again, it's going to be carnivores and scavengers, so you just kind of have to think, well, what are the carnivores and scavengers out in that area? Tend to be these hyenas. And the sylvatic frigid is Trichinella nativa, right? And that's up in the polar regions where we know that they go into polar bears, believe it or not. So again, carnivores and scavengers, all right? Carnivore consumes it, and then scavengers can pick it up at that point. And again, humans can get infected if we eat undercooked meat. Cook your meat. So yeah, know these terms: sylvatic temperate, sylvatic torrid, and kind of have ideas of where they would occur. I'm not going to ask you what life cycle patterns could Trichinella spiralis be showing. We use Trichinella spiralis as our life cycle example, and we're going to talk about its pathology and stuff. And we know a lot about it because it affects humans. All right, so our nurse cells are living cells. That's what they are. And it's these living cells that are housing our J1s. Now, the cells that serve as that can function as a nurse cell are the striated muscle cells. And what our worm is going to do is completely remodel it so that it now acts to protect the worm, protect it from the host immune system, and nourish the worm. All right. So how does this happen? How do we get the conversion? Well, I've got a diagram here to kind of show you just the general pattern. And this is days. So at day 0, the worm penetrates, burrows into our muscle cell. And when it does that, it can cause damage. So you might feel some of the pain. And then once it's in that cell, it induces this change that causes our cell to enlarge. And ultimately, our worm's going to enlarge too, and it forms this cyst. Now, these changes are all promoted by the stichocytes, by secretions of the stichocytes. How do we know that? Because you can take these stichocytes out. You can isolate the secretions, and if you inject them into striated muscle cells, you see these changes without having the worm inside of it. So we know the stichocytes are causing this. What are the changes? Well, if we're remodeling the cell and making it so that it is going to protect and nurture the worm, then we'd have no use for that cell to actually function as a muscle cell. So the myofilaments are going to be lost. Right? What's the purpose? They're not using it. And the worm doesn't want them to use. The worm wants that cell to its own purpose. Second thing that it's going to happen is that the cells are going to be maintained at that G2m phase of mitosis. What is that phase? That G2m phase.
Proliferative things.
Good, proliferative phase. And you remembered that, right? Yeah, so if you go back, this should probably be cell bio, maybe genetics. But you know, during the cell cycle, you've got a lot of things happening, and in preparation for that actual mitotic step, that M phase, you've got a lot of things happening, a lot of replication, growth happening, enlargement so that the cell can actually divide. This phase is basically right before that period where now our chromosomes start aligning, separating, and then telekinesis happening. All right, so we are at our proliferative phase of the cell. But it's not just there. So like in normal mitosis, your chromosomes replicate. So you get 2 times 2n diploid number. All right. And then when it splits, it gets back to your 2n number. It's not here. Trichinella actually causes it to divide one more time, so you actually have 4 times 2n. So instead of like our nuclei dividing once and then separating, it divides twice, and the nuclei remain enlarged. Why is that? Well, we're going to have that cell produce proteins to help nourish the worm. All right? If you only have 1 copy of a gene, right, you're only making 1 protein. If you've got 4 copies, you can make 4 proteins at the same time. So it's probably tied to this protein production. If you increase the amount of DNA that you have in there, you increase the amount of proteins that can be grown. Also with this, definitely the protein production increases at this phase. So myofilaments are lost. Our cell is transitioned to this proliferative stage, increased protein production. We also have upregulation of collagen synthesis genes. All right, so what we're doing now is if we increase collagen synthesis, we're starting to form a capsule around the cell itself, around the worm. All right, so we're going to encapsulate this worm in collagen. And we have cross sections. We've got slides of cross section of nerve cells. You can see the collagen in there. All right, why is that good? Collagen produced by the host isn't going to be harmed by the host immune system. So by encapsulating our worm, we're protecting it from the host immune system. Problem though is that when you encapsulate it, you wall it off from the outside, so it's not going to be fully encapsulated. It's not going to be impermeable, right? But it's definitely going to be less permeable than if it wasn't encapsulated. So for that reason, then these cells actually upregulate vascular endothelial growth factor, VEGF. What is VEGF? It stimulates angiogenesis. What is angiogenesis? The production of blood vessels. All right, production of blood vessels. So it's going to form these blood vessels that surround the nurse cell. So we've already made it less permeable to nutrients. Now we're increasing blood flow around that area in the hopes of offsetting some of that impermeability. Has this been used in the medical world? Yeah, this has been used. If you have someone that has a blockage and they need more blood vessels to grow, they've stimulated, they've secreted this VEGF to try to promote blood vessel formation in some of those areas. So this worm, when I was in college, or right before I was in college, this worm was a popular research topic. How can we utilize the stichocytes and their secretions to try to help with limited blood flow in humans? Did you guys get this? All right. So speaking of VEGF and stuff, all right, so encapsulation presents a problem. Right? It's going to limit nutrient acquisition, gas exchanges and stuff. All right. Now the VEGF is kind of the counter strategy. It's a strategy to counter that encapsulation. So this VEGF is going to promote blood vessel formation and cause a network of blood vessels to form around that nurse cell. That network of blood vessels is called RETE, R-E-T-E. All right, and this would be a schematic of it. All right, so you've got your nurse cell, you've got the collagen encapsulating it, and then you have this network of blood vessels surrounding it. This is a RETE. It's actually an interesting story on how they found it, right? But ultimately, they kind of had it crystallized and dissolved the content, so it left this cage of blood vessels. Pretty cool. I think I have a book that tells that story. All right, so what about these RETE? The size of the RETE, the size of these blood vessels are actually larger than the size of capillaries. So, our body has capillary beds. We've got capillaries in the lungs. We're gonna see that. We're gonna talk about those. Right? But the size of these things are actually larger than the capillaries. And there's benefits and disadvantages to this. The benefit is that because they're so large, because they're able to move blood, you can have very good transport of nutrients and waste products. So nutrients into the cells, taking waste away from the cell. So you can get very good transport of movement. But the downside is associated with gas exchange. All right? So if you have a large blood vessel, gas exchange is actually hindered because the partial pressure of oxygen at that point won't ever get to the level where oxygen will now become disassociated from the heme group and hemoglobin. So you go back, I'm sure you talked about it in Bio 1, 1406, maybe? The dissociation curve, partial pressures. Biochem probably talks about it, right? Some of you that have had it.
Do you remember it in bio?
In general bio? Yeah, I think that's where we talk about it. You probably also talked about like alternatives to hemoglobin.
Oh, yeah.
So horseshoe crabs, use copper, blue blood. Blue blood, that's kind of cool. Or deep sea,
Isn't that the same thing with octopus as well?
ocean mammals,
Blue blood.
possibly. I know horseshoe crab. Again, a physiology course in grad school, we drew some blood. Horseshoe crabs. Pretty cool. Anyways, yeah, we don't ever get low enough to where oxygen can be released. And if we don't get to that point where oxygen is being released, that means we're not going to have oxygen going into that cell. All right, so what's the response to this? It goes back to our life cycle. Our worm's now going to shift towards anaerobic respiration. Does it loses out on the efficiency of ATP production because we're not using oxygen as that in the electron transport chain as our electron acceptor, electron receiver, but we don't really need to because we're not actively growing, we're not actively reproducing. We're basically, the worm is just in this place to survive until the host gets consumed. So it only needs to make enough ATP, enough energy to maintain its life. So, yeah, the reading, great for nutrient flow, great for waste flow, get it in and out of that nurse cell, but you're not going to be able to get oxygen, so our worm shifts to anaerobic respiration. And then once we get released, hey, yeah, start going back to oxidative phosphorylation. Questions? We will, all right. Here's our diagram. When I said it's moving through the gut mucosa, that's what's happening. Here's our luminal surface up here. All right, our worm's kind of just burrowing through the cells in the cytoplasm. So we've got essentially 3 phases of our infection, or at least 3 phases when we might see pathology. All right? So if you get Trichinella, you're going to exhibit a disease called trichinosis. The first phase is tied to these juveniles and to the adult stages as they're moving through the gut mucosa. All right, that could produce symptoms. Now, often it's undetected. If you only have a couple worms, or a small number of worms, you're probably not going to notice it. All right, but if you just had a undercooked pork sausage, right, that had a bunch of Trichinella in it, you're probably going to have hundreds of these things moving through it. All right, so as they're moving through these, we are causing damage to the gut mucosa itself that then induces inflammation. All right, but it's not just the damage. We also have the waste products because our worms are feeding, producing waste, which is now stimulating the host immune system, and the primary response is going to be inflammatory in nature. So our symptoms are all tied to increased inflammation, which would be nausea and sweating and vomiting and diarrhea. Diarrhea comes from irritation of the gut, inflammation of the gut. All right, we do have the possibility of secondary infection. So say you could have bacterial infection that develops. So bacteria gets into that open wound and then spreads, and you go to the visit, you're not feeling good, and they say, okay, here's some antibiotics. And you get it cleared up because normally by the time you go see a doctor, you get the antibiotics, and you start your treatment, that's about the time when our adults have finished reproducing and releasing J1s, which is kind of marking the end of this stage. So we start losing these types of symptoms. Now what we do see, and this is kind of a clue that it was Trichinella, that at the end of this phase, which is about like 5 to 7 days after you first start showing these symptoms, you tend to exhibit facial swelling, facial edema, and then fever. So again, if you think about seeking medical attention, seeking medical treatment, you're going to get it. You're going to get antibiotics maybe on the 3rd or 4th day. All right, antibiotics will make you feel better, but it's not immediately. You have to give it a day or two to kick in. And at that time, yeah, you might have your last bit of fever, you might have some facial swelling, but would you necessarily recognize it? Now you do. Now you do. You'll probably forget this part. You'll remember the pictures of the pathology.
My friend had a black bear in Alaska. He ended up getting Trichinella from it.
Yep.
And he actually gave me some of the meat. I guess he didn't cook it well, but how did the bear get it? I guess he eat domestic animals for a while.
How did the bear get it? Was it up in Alaska?
I think it was either Alaska or was it in Washington?
Yeah, it's probably this. Carnivores, the sylvatic temperate. So bears, they're carnivores, scavengers. So yeah, the bear possibly ate another bear. He didn't cook his meat. He must like rare steaks.
I think when they shot it, they cooked it right after they killed it, basically.
Yeah, they didn't.
And they were up in the mountains that had they were just not paying attention or what. They didn't cook it, but they gave me some. He didn't develop it. He didn't notice it until a while after.
What was the sign that
That
he noticed?
I don't remember him telling me what it was. They started going real bad. And I think one of the guys there was they started having like real bad headaches. Finally went to the doctor and they said they had Trichinella. So.
Crazy. He's gonna have it.
Yeah.
And bummer. But there are probably worse things that could have happened.
Oh
Definitely.
You can treat it though, right? Like, get rid of it.
You can treat it. As we'll see, I think there are worse nematodes that they could have gotten.
yeah. Oh yeah, for sure.
All right, so that's phase 1. Phase 2 is associated with the juvenile stages migrating. So remember, with our life cycle, our adults mate, they release the J1s. The J1s travel in the circulatory system, and they don't know where they're going. Right? They just up and decide, I'm going to burrow out of the blood vessel and start penetrating, hoping to find skeletal muscle. All right, so this is our second phase. So what do we typically see? Well, anytime you have damage to the cells and any of our cells, we're going to get inflammatory response. So what we can see is localized edema, localized swelling because of damage to blood vessels. Also get some localized swelling in various parts of the body. And then the other symptoms that you get all kind of depend on where those worms are penetrating and burrowing. So it could be pneumonia that you end up getting because they burrowed out of the capillaries and they're burrowing through the lung tissue. You've got inflammation. You're going to have increase in fluid in the lungs leading to this pneumonia. Chad is your friend, right? The headaches. It burrowed out of the capillaries of the brain. It's probably causing this encephalitis. Some brain swelling, inflammatory response, and the swelling is what was alerting him, hey, I've got these headaches, they're not going away. Maybe I need to check it out. Deafness, if the nematode gets out and burrows through the inner ear, burrows through an eardrum. Has it happened? It's on the side, so it's definitely happened.
They avoided that one.
Yeah, right? Eye damage. Gets out, burrows through the retina, damages the retina. That's permanent damage. You can't fix it. Same thing with the eardrum, although implants, is it cochlear implants? No, could save you. I don't think they have eye implants yet. All right, so yeah, just various types of symptoms, just kind of depending on where they're at. All right, you can get death from these migrating juveniles. So this is normally it happens from heart inflammation, so myocarditis. So it burrows out, gets into the cardiac muscles, burrowing through, causing damage and inflammation up to the point where there's so much inflammation it just causes the heart to stop. All right, so they move through, but they don't insist. Popular site. Yeah, that's later. All right. You ready for that? Next one. So the third phase is nurse cell formation. All right. So this is typically 10 days after the first symptoms of trichinosis. So I'm curious if your friend exhibited any of these.
He said it was about, I think, like 2 weeks after they started having it. They said they just couldn't get out of bed. They were so sore and like achy. And they had headaches and stuff, but I don't.
Sore and achy? Yep, probably this. Intense muscle pain.
Yeah, there you go.
All right. I had in my notes I say, yeah, feel the burn. Yeah. Some of you have been in weight rooms and heard people, those phrases, feel the burn. This gives new meaning to that. All right, because the worm burrowed into your skeletal muscle and is now changing it. So you can feel it. I mean, it'll hurt. You can have potential breathing problems, and this is when they insist in the diaphragm. So the diaphragm, right, is negative pressure breathing. All right, you change a muscle cell so it's no longer functioning as a striated muscle cell. You can cause breathing problems. Another popular site is getting into the jaw muscles. So you could often see you could say the masseter muscle or the masseter muscle. I think it depends on who you have for anatomy and how you pronounce it. So masseter muscle, this is kind of important because this could lead to a misdiagnosis of mumps. And someone comes in and says, oh man, you might have mumps. Now, if you don't tell them any of the other symptoms that you've had, they might just treat for mumps, but make sure you tell everything. And then you can have death. And typically, if death is going to happen, it's from heavy infections, and it's from all the inflammation that's happening that the body just ultimately just kind of starts shutting down. So heart failure, kidney malfunction, respiratory complications. You know, it might not just be one of these. Maybe it's 2 or 3, or maybe it's multiple things. Now, there are other symptoms, other triggers, other things that kind of suggest you might have trichinella. One of those is the splinter hemorrhages. So you can kind of make out, you've got these ruptured blood vessels because the worm burrows out of the capillary, causes them to rupture, and then you're going to have slight bleeding up underneath the nail bed. Yeah, slightly better picture here. Head. Let's see if I can open in the window. Yeah, that's better. Splinter hemorrhages kind of makes it look like it's a splinter underneath the nails. All right. Sometimes you get it. I mean, I've had it. Smash your finger in a door or something like that. You can have hemorrhages under the nail bed. But these things, again, coupled with maybe muscle pain, maybe with some fever, you might have trichinella. Other is this conjunctivitis, and periorbital edema. These are the worms that have kind of busted out the capillaries around the eye, causing swelling and inflammation. So you get swelling around your eye. It looks reddish, and there are several pictures that you can find on the internet. These were both pulled from trichinella.org, an actual website dedicated to trichinella. So, all right, questions? All right, so we have trichinella slides. We've got nurse cell slides that we can look at in the lab. Monday has looked at them. Wednesday, you guys are going to be looking at these. All right, so next up, we're going to move to the GI tract residents. So we'll start with the first batch, which will probably be hookworms only. There's a lot to discuss with hookworms. All right, so we'll pick that up on Friday. If you have questions, please email me and cook your food.